Hello, general biology students. Dr. Martin. This is chapter 10, and this is the 11th week of the semester. I hope you had a great break last week in spring break. I had a great spring break. Spent a lot of time with grandkids, and I hope you had a very productive spring break as well. But hey, things begin to happen really fast here as we get past spring break. It always does. Your schedule, other than school, gets busy. My schedule gets busy, and but hey, let's give a lot of attention here. This is the last chapter before our exam next week. So next week, week 12, is just an exam, some review, and maybe an assignment, and that's it. But that'll be your second unit exam. So 2/3 of the way done next week. So here we are, chapter 10. This is the last chapter that we're going to study here in unit 2. Chapter 10 is on patterns of inheritance, chapter 10. And it's really about genetics. Genetics is a science that explains and predicts patterns of inheritance, traits that are passed from parents to offspring, and how we can predict them. Now it's kind of fun to use neat traits, hair color, eye color, things like that, but this is also very important when we're talking very serious traits. Some of them, very serious inheritable traits that can be very life-threatening, and so it is a very serious chapter too. But it's kind of fun. But it's fun to follow along. It's not a real long chapter, but be sure you pay attention to the details. So when we talk about genetics, we're talking about genes. Gene is a segment of a DNA molecule or a chromosome. A gene is a small segment of a chromosome that has the instructions on it to build something, usually a protein. It codes for something. Remember, you're about 50% protein. And in order to make proteins, we have to find the code or the information on genes that are on your chromosomes. Hey, look at all these people in this picture here, all different colors, all different skin colors, height, gender. Hey, I was looking. I see only one bald guy on there, but different hair colors, and but so diversity now. And again, we tend to humanize this. All organisms have genes that code for specific things in their body function, their body structure. So we're going to humanize it a lot because that's where the interest is. But remember, all organisms, from amoebas to bacteria to humans, have genes on their DNA. And this is the science of genetics. And so just a little bit of a review. Recall this, that chromosomes, now we're not going to talk much about chromosomes. We've done that already in previous chapters. But for the human genome, we have 46 chromosomes. Here's a karyotype. Remember that from the last chapter. This karyotype lists or displays a person's chromosomes here. But what we're going to talk about in this chapter is not whole chromosomes. We're going to talk about specific sites on chromosomes where genes are located. In fact, we call these loci or a locus. It's a location where a specific gene is located that codes for something. Remember, we talked about the Human Genome Project last week and how President Clinton in the 1990s initiated the Human Genome Project, and we actually identified every chromosome and every gene on every chromosome of the human genome and made it public for anybody in the world to use on the internet. A person who has two sets of chromosomes, we call those diploid. One set of chromosome is haploid. Remember what kinds of cells have haploid and what kinds of cells have diploid sets of chromosomes. Gametes, pollen grains, spores, they have sex cells, they have haploid numbers of chromosomes. A zygote, fertilized egg cell, sperm cell, body cells, they have diploid number of chromosomes. You received half of your chromosomes from your mother, half from your father. Here's chromosome number 1. You received 1 from your mother, 1 from your father. Here's chromosome number 22. You received 1 from your mother, 1 from your father. Even the sex chromosomes, you received 1 from your mother, 1 from your father. So remember the terms diploid and haploid as we go through this, but we're going to talk a lot more about genes than we are chromosomes. So remember, these are homologous pairs. So you have two number ones, two number 2 chromosomes, two number 8, two number 22s, two sex chromosomes. They are twin chromosomes. They are homologous, homo meaning same. In other words, these two chromosomes, number ones, they have the same genes and that code for specific things in your body. So not only do you have two chromosomes for your in your cells, each contributed by your mother and father, you also have two genes for every trait about you. There are two genes. One was contributed by one parent, one was contributed by the other parent. And we're going to talk a lot more about genes than we are chromosomes. Technically, these genes on these specific chromosomes are called alleles. And sometimes we learn, we use these terms interchangeably. But let's talk about this just for a second. We know that there are some very obvious genes for eye color. Eye color is terrible to use because it tends to change over time, but it's kind of fun to talk about in humans. As you would suspect, dark eyes, brown eyes are a dominant gene trait. Blue eyes are kind of a recessive gene trait. Well, these genes can be the same or they can be opposites. Okay. In other words, you may have different alleles of the same gene, different types of the same gene. You may have brown eyes, but you may have a blue-eyed gene that's simply being masked or covered up. These are called alleles. They can be identical in the two pairs of genes, or they can be different in the two pairs of genes. Like these two genes on the pink chromosome, these two A's are the same. Notice they're capitals. These two B's are the same. They're capitals. But look at here. These two genes D's are small, lowercase. Okay, so you have two genes for every trait about you. At least two genes for every trait about you. Gregor Mendel was an Austrian monk in the 1700s. He didn't have the ability to know what genes were. He didn't know what cells were, but he was a strong scientist. He was a geneticist, what we call the father of genetics, the science of genetics. And I mean, we're talking a long time ago, but he didn't even know what genes were, didn't even know what cells were, didn't know what chromosomes were. But he cross-pollinated pea plants in his garden as a monk. And he began to notice that there were different traits in pea plants, just like hair color, eye color in humans. He noticed different traits in pea plants. The height of a pea plant, one tall, one short. Colors of seeds, like all green here versus seeds that are yellow. He would cross-pollinate, and he began to be able to predict what offspring would look like. Remember, that's the science of genetics, predicting patterns of inheritance. And so he could predict what the offspring would look like when he crossed the pollen. They used paintbrushes, and that's how you can cross-pollinate plants. Instead of allowing them to self-pollinate, you can cross-pollinate using paintbrushes and pass pollen from one plant to another, and you can cross-pollinate that way. And he was able to predict what offspring looked like. For instance, some clues here. He knew that if a plant with green seeds pollinated itself, it produced green seeds. Plants that had yellow seeds, if they self-pollinated, they produced yellow seeds. But he also noticed that there were some yellow seeded plants that if he allowed them to self-pollinate, every now and then they would produce a green seed, what we call a hybrid. And so he began to predict what offspring would look like in these, what we call crosses. We know this, that some alleles or some genes are dominant. In other words, if they are present, they are expressed outwardly. If they're present, you see them. If you have a dark hair gene, brown hair, black hair. It doesn't matter if one of the genes or both of the genes. If it's there, you have black hair, brown hair. Dark-eyed genes. Even if one of the genes is a dark-haired gene, it's expressed. That's what we call a dominant allele or a dominant gene. For instance, if he pollinated a green seed plant with a yellow seed plant, yellow seeds. That's the dominant gene, the dominant parent color for seeds in pea plants. But every now and then he would cross a green seeded plant with a yellow seeded plant, and he would get a mixture or a hybrid of colors of seeds. Now I know you're smart enough to know what's going on here. Okay, here we have this parent can only pass on yellow seeded genes, and but over here it's a little different. So what we're going to do is we're going to show you how we can predict patterns of inheritance. We're going to use pea plants. It's a little more fun using human traits, but there's so much variability in human traits. We have to be careful with that. So we know what a dominant gene is, but he also realized what a recessive allele was. Now, he didn't know what a gene was. He called them factors. He didn't really know what else to call them, but he was way ahead of his time in predicting what offspring would look like. For instance, here's that same cross, a green seeded plant with a yellow seeded plant. If this cross occurred, the majority of offspring would have yellow seeds, or at the very least, half would have yellow seeds. He was able to identify that in pea plants, yellow seeded plants was the dominant seed color. Green seeded plants was the recessive color. In other words, it's hard for it to show up. It can only be expressed if both genes are recessive. In hair color in humans, blonde, red, they can only be expressed if both genes are recessive. No black, no brown genes. So in dominant genes, just one of them has to be there in order to show up. But in recessive genes, both of them have to be recessive in order for them to show up. He found this out in pea plants in using their color. A couple of other terms we need to talk about. There's what we call a genotype, and then there's what we call a phenotype. A genotype is what the two genes for that trait are in your genome. In this particular plant, both genes are dominant. We know that because we have capital Y's. See that? Yellow, yellow. Okay, so this is a plant that is homozygous. Both of them are the same. Dominant, dominant. Remember, there's two genes for every trait. Look at this one here. This plant is yellow, but it is heterozygous. Hetero meaning different. Capital Y, lowercase y. Dominant gene, recessive gene. Remember, two genes for every trait. But if the dominant gene is there, it is expressed. So this is a yellow seeded plant. But look here, this is a homozygous, homo meaning same, recessive. Lowercase y, lowercase y. In this case, it's the recessive trait of green seeds. Remember, in order for a recessive trait, blonde hair, red hair, in order for it to show up, both genes have to be recessive. Homozygous. Here, homozygous, both of them dominant. Heterozygous, one dominant, one recessive. These are what we call genotypes. This is what the traits look like on genes. Over here are the phenotypes. Phenotypes are what they look like outwardly. How are they expressed physically? How are they expressed on the pea plant? How are they expressed in eye color, hair color? Okay, homozygous, both dominant, yellow seeds. Heterozygous, one dominant, one recessive, yellow seeds. Remember, dominant, if only one gene is there, it's expressed. Here, homozygous, green. Green, yellow, black hair, red hair, blonde hair, blue eyes. Those are phenotypes. That's how they are expressed outwardly. When we look at the genes on the chromosomes, that's genotype. That's how they show up on the genes. Okay, so you know the difference now between a dominant allele and a recessive allele. And now you know what a genotype is versus a phenotype. And you also learned a couple of terms, homozygous versus heterozygous. Important terms for us to use in genetics. So when we cross two parents and we try to predict their offspring, we use what's called the Punnett square. You might remember this from high school biology. These in genetics, if you took high school, everybody takes high school biology. So this is what we call a Punnett square. It's a nice way for us to predict what offspring are going to look like. For instance, here's one parent here. If we put the female here at the top of the square, so here's a female parent, pea plant, yellow seeds, but look at here, it's a capital Y, lowercase y. That means we have a heterozygous genes, okay, one gene from the father and then one gene from the mother. One was dominant, one was recessive. Remember, if the dominant gene is there, it's expressed regardless. Here's the male parent. Now, pea plants don't really have moms and dads, but they have pollens, and then they have stigmas. They have reproductive parts. So here's the female parent. Here's the male parent. Look, the genome is the same. The genotype of these two parents is the same. They have yellow seeds. Okay, yellow seeds. Notice the genotype is uppercase, lowercase, heterozygous, but they have yellow seeds as their phenotype. So what we're going to do is we're going to cross these two plants, and this is what Mendel did. He'd get a little paintbrush, and he would brush pollen from one plant to the next, and he would fertilize these two plants. So we're going to do a cross now between two parents. Now, remember in meiosis, these two genes separate. One goes to one gamete, the other one goes to another gamete. Remember, not only do homologous chromosomes separate during anaphase of meiosis, genes separate as well. So this parent, you as a parent, you contribute one gene for every trait about you to your egg cells, to your sperm cells. You don't contribute two, you contribute one for every trait. Okay, remember diploid and haploid. So this parent right here, this female, is going to pass on a dominant gene for seed color, and it's going to pass on a recessive gene for seed color. Here we are right here. Capital Y, lowercase y. Hey, this parent here, the same, is going to pass on a dominant gene to half of the gametes, recessive gene to half of the gametes. And here's how we do it right here. Now they've depicted sperm cells here, as if it were humans. But in plants, it's technically pollen. But so here's a parent here that has yellow seeds, a parent here that has yellow seeds. That's their phenotypes. Their genotypes are capital Y, lowercase y. And so here's what we pass on. One gamete passes on a dominant gene, one passes on a recessive gene. Same way up here, dominant gene, recessive gene. During fertilization, we show all possibilities that may potentially occur. Potentially, these two gametes will join together in fertilization. If that's the case, we have two dominant genes. This is a yellow-seeded plant. There is a possibility that this and this gamete will fertilize. In that case, we have a dominant and a recessive. Still, dominant genes produce dominant colors, yellow seed color. There is a possibility that this gamete will join with that gamete. Again, heterozygous, dominant, recessive. We always write the dominant one first. But look right here. This is where Mendel began to scratch his head. Two yellow-seeded parent plants produced potentially a green-seeded offspring. You see how? Recessive gene joins with recessive gene. So there's a potential in this cross that we could have green seeds. Kind of head scratching here. So the genotypes are out of 4, so it's 25%, 25%, 25%, 25%, or 1/4, 1/4, 1/4, 1/4. So genotypes, 1/4 will be this, 1/2 will be this, 1/4 will be this. It's a 1-to-1 ratio. You see that? But look at the phenotypes, the actual colors. 3 out of 4 will be yellow seeded plants. 1 out of 4 will be green seeded plants. A 3-to-1 ratio. I'm sorry, my ugly mug's in the way here. So this is what we call a Punnett square, and this is how we predict offspring in organisms, crosses between organisms. In this particular example here, and this is the same example, there's nothing different here. You understand what's going on here. We call this a monohybrid cross. It's a cross between two organisms involving one trait, one single trait. If we could do it in humans using eye color, monohybrid cross. If we could do it in humans using hair color, monohybrid cross. If we could do it in humans using 2 traits, head color and eye color, you would call that a dihybrid cross. We could even do trihybrid crosses. They get quite complicated, but we're only going to do what we call monohybrid crosses, crosses between 2 parents involving 1 trait. So let's talk a little bit more about this, and this is what we call a Punnett square. It's just an easy way to predict the ratios of offspring. If I could give you some practice problems for you to do this, and maybe on the quiz, could you do some monohybrid crosses? Could you cross? This is easy here because we have 2 what we call heterozygous parents, but what if we had other types of parents here, other types of genotypes? Could you do a Punnett square and predict the offspring? Maybe this week we can practice that. I'll give you some practice problems here. So these answered a lot of Mendel's questions, and so he developed 2 Mendelian laws. The first law was that for every trait about an organism, there are 2 genes that code for that trait. His second law was that somehow during the fertilization process, that each parent passed on 1 gene to their offspring, or to their gametes, or to their sperm cells, egg cells. So what we call law of segregation. And so these are the laws that Mendel developed. And so the main thing for you to understand is understand what dominant and recessive is, understand what genotype versus phenotype is, and can you do a Punnett square cross? They're kind of fun, and I'll give us some this week to kind of practice a little bit. In fact, I think there's one here. Yeah, here's a good one to practice here. So if we had in fact, this is a very good question here. What is the genotype of a plant grown from a yellow seed? Well, it could be 2 possibilities. Okay, both of these are yellow, but look, this one's homozygous, this one's heterozygous, but they're still yellow seeded plants. So what if we had a parent that had 2 dominant genes? We cross-pollinated with a parent that had 2 recessive genes. Notice it's green. That's the only way you can have a green seeded plant is if both genes are recessive. So if we did a Punnett square where we segregated out a capital Y, small y, lowercase y, and then we did our crosses here, and here, and then here and here, and then we do here and here, all possibilities, and then here and here. You see why we do a Punnett square, so we can show all possibilities. Notice these are gametes, one gene per trait. These are offspring, 2 genes per trait. Can you see that every single offspring is going to have the dominant color? They're going to have a dominant gene in their genome, therefore they're going to have the dominant color in their phenotype. 100% will have yellow seeds. Can you do this cross down here? Now I've covered up accidentally. I wish I could, in the presentation I could move my mug here, but I can't. But what if we did this? What if we had a heterozygous yellow seeded plant? Again, a green seeded, both recessive. What if we did this cross and set up our Punnett square? These are organisms, 2 genes per trait, but this parent can pass on a capital Y, lowercase y. This one can only pass on one type. And then we do our cross. You see what's going to happen here. We're going to have about 50% yellow, 50% green seeded plants. That's phenotypes. The genotypes are a little bit different. We're going to have 50% heterozygous and 50% homozygous recessive. So you can do some Punnett squares, monohybrid crosses, Punnett squares. That don't get any more complicated than that right there. And this is just a little bit more of an explanation of what we did on the previous page. Now, that's how most genetics works. It gets a little more complicated, as you might imagine, but this is general biology, non-majors biology, so we're not going to get any more complicated than that. But let's show you some exceptions to these traits, some exceptions to Mendel's laws. For instance, there are some flower types that if you have a if you have a red colored plant, flowered plant and you crossed it with a white flowered plant, you get pink flowers. Now this is very similar to melanism in humans. There's not just black or white in skin color in humans. All across the globe, there are all different shades of melanism in skin color. Well, there are some traits that don't come out just black and white or red and white. Sometimes there's a blending of colors or genes. And so in other words, there's no dominant, there's no recessive. They are co-dominant. And that's what happens in these kinds of genetic traits. The human blood type and human skin color are both co-dominant traits. You know this. Some of you may be type A blood types. Some of you may be type B blood types. But if a parent that is type A has a child with a parent that is type B, AB blood type results in the children. And in some cases, neither of these are present, and we have a type O blood type. So in other words, both genes are dominant. It's what we call co-dominance. Skin color is like that as well. In fact, in skin color, there's not 2 genes. There's as many as 6 genes for melanism content in skin color. And that produces the multiple shades of skin color across the globe. So co-dominant traits or co-dominant alleles, they don't cover each other up. There's no dominant, there's no recessive. They're just all dominant. They're all expressed. And the human blood type and human skin color are classic examples. Another exception to the Mendelian traits are what we call sex-linked traits. These are traits that are found on the X chromosome. Now, we're talking genders here, so we have a female with an XX genotype. Then we have a male that is an XY genotype. Now, you and I both know that in about 1 in 700 births, there are some abnormalities. You remember we call those aneuploidy or polyploidy situations in sex chromosomes. But under most conditions, the majority of the time, an individual has an XX genome, and if they do, they are female. They may have an XY genotype, and if they do, they are male. If we did a Punnett square, okay, here we could do a Punnett square for sex or gender in humans. Look here, we have a female that can pass on one gene or chromosome to her offspring, and she can only pass on an X chromosome. But look here, in males, males can pass on either an X chromosome or a Y chromosome to their gametes. Remember, 2 genes in our body cells, only 1 gene in our gametes. And if we do a Punnett square, here we go. This potential egg cell will form with this sperm cell. In that case, we have an XX female. Here we have XX female, and then you can see what's going to happen down here with the Y. So now you understand why half of the human race is 50% male, 50% female. Now, if a person is pregnant, what is the chances that their offspring will be male versus female? Well, it's 50-50. What if a set of parents have 10 children, all of them males? What are the chances that the 11th child will be male or female? 50-50. Even if you would think that the chances are stacked against them. But now, what are the chances that all 10 or 11 would be male versus female? Well, those are much less. But the chance that that next child will be a male or female, 50-50. Now you understand why, is because what we pass on in meiosis into our gametes. Some genes, though, are linked to the X chromosome. And if they're found on the X chromosome, but not the Y chromosome, then there's nothing on the Y chromosome to mask it or dominate it. These are what we call sex-linked traits. And the X chromosomes always carry these traits. Baldness, pattern baldness in humans is a sex-linked trait. Red-green color blindness in humans is an X-linked trait. Hemophilia in humans is an X-linked trait. In other words, if a dominant gene is on the Y on the X chromosome, it's going to show up. If the recessive gene is on the X chromosome, it's going to show up because there's no other gene on the Y chromosome to mask it or dominate it. And so these are what we call X-linked traits, or sex-linked traits is what we call them. And so it's hard. It was obviously Mendel couldn't really predict those. And this is kind of what is explaining those traits. Both of these slides here. We're pretty much done here, but what we call sex-linked recessive disorders are some of them are fun to talk about. Pattern baldness, red-green colored blindness. We have a red-green color blindness in my family's lineage. But some are very serious. Hemophilia is a very serious X-linked trait. There are others, but there's nothing on that Y chromosome to dominate that trait. So, and then here's the skin color trait. Again, not only is it a co-dominant trait, it is a polygenic trait. In other words, Mendel said 1 trait, 2 genes. But we now know that there are some traits that have multiple genes, polygenic traits, and skin color, melanism production in skin color is one of those. So anyway, so that's Mendelian genetics. Kind of fun to talk about, kind of interesting to talk about. I want to make sure you can do some Punnett square combinations. I'll even send you a few practice problems this week, and you can practice them because next week is the exam for this unit 2, chapters 3, and then 8, 9, and 10. So be prepared for that next week. Take good notes on these slides for chapter 3, and then 8, 9, and 10. Have a good week, and we'll prepare for next week.